Well, hello there and welcome back to our series of videos on the topic of molecular genetics. Now, in the last video, we stopped at this idea that the genetic code is a triplet code. So genes, which are a sequence of nucleotides, get converted to polypeptides, which are a sequence of amino acids. And what helps you to convert nucleotides into amino acids is the genetic code. And we saw that in this genetic code, right, nucleotides are grouped into triplets called codons, and each triplet, each codon codes for one amino acid. So this is a big idea. But what is the process? How does it happen? How do codons become converted into amino acids? That's what we'll explore in the rest of this video. So it turns out that polypeptides are made, or genes are expressed, through a two-step process. And the first step is called transcription, and the second step is called translation. So in transcription, what happens is that the gene, the DNA template, is used to synthesize a strand of RNA called the messenger RNA. So you can see the messenger RNA over here. Now this messenger RNA is complementary to the DNA template. So you can see that when there's a T on the DNA template, the RNA has an A. However, you will notice that this messenger RNA contains a base called uracil instead of thymine. So RNA does not have thymine, it has uracil instead of thymine. More on that later. Now in the second step, this messenger mRNA is then translated and used to make a polypeptide. So from this mRNA, we end up with a sequence of amino acids, which is a polypeptide. So what is this RNA which we talked about in the last slide? Well, RNA is quite similar to DNA. It's also a polynucleotide. It's also made up of nucleotides, but it has some differences. Firstly, in DNA, remember that in the nucleotide for DNA, so here's our nucleotide, phosphate, sugar, and base. In DNA, this sugar is a deoxyribose. But in RNA, this sugar is a ribose. Right? So in DNA actually is a deoxyribose, meaning that it lacks an oxygen somewhere. But in RNA, the sugar unit is ribose. The second difference is that in DNA, this bases comes in four forms: A, T, G, and C. But in RNA, this base comes in four forms as well, but instead of thymine, we have uracil. Thirdly, in DNA, usually DNA molecules are double-stranded, so they're made of two strands which are held together by hydrogen bonds. But in RNA, it can be either single or double-stranded. So for example, your messenger RNA is single-stranded. Now because DNA is double-stranded, the ratio of A and T and G and C is always 1:1. So it's always the same number of A as T. But because RNA can be single-stranded, there's no fixed ratio between A and U or between G and C. Okay, so how are genes expressed? So let's take the example of this gene over here. It's a segment of DNA and I'm going to enlarge it for you. So this gene, you can see the sequence of nucleotides here. It's made up of two strands of nucleotides and there's complementary base pairing between the two strands. Alright, so in the first step, when a gene is expressed, is that the two strands of the gene will separate from one another. They will unwind. So here we go. The two strands of the gene unwind. Then one strand, we'll call it the template strand, and the other strand, we'll call it the non-template strand. So only one strand of the gene will act as a template strand. And this template strand is important for transcription. Okay, so during transcription, the template strand is used to synthesize a strand of messenger RNA. So let's see that happen here. So here's my strand of messenger mRNA. And you can see that this strand of messenger mRNA is complementary to the template strand. So where the template strand has T, the mRNA has A. Where the template strand has A, the mRNA has U. Right? Remember that RNA has uracil instead of thymine. Where the template strand has cytosine, the mRNA strand has guanine, and so on. It's complementary. So one important thing to note is that the sequence of the mRNA is actually the same as the sequence of the non-template strand, except that we replace thymine with uracil. So that's why we also call the non-template strand the coding strand. because you can see that the codons on the non-template strand actually match the codons on the mRNA. So the mRNA is really actually carrying the information from the coding strand. So it's really the coding strand that carries the code for the polypeptide, which the mRNA looks like the coding strand, right? But in order for the mRNA to look like the coding strand, it needs to use the template strand to synthesize itself. Right, so that's why this is called the template strand, this is called the non-template strand or coding strand, or one more name that people give it is it's called the sense strand, because it makes sense. Okay, so importantly, transcription takes place in the nucleus, because that's where your DNA is found, that's where your genes are found. Transcription will take place there and produce the mRNA. Then the mRNA will move out, it will carry the message from the genes and move out of the nucleus through the nuclear pores. It will move into the cytoplasm. And there, translation, which is the second step, will take place at ribosomes. Now remember, ribosomes can be found either on the rough endoplasmic reticulum or can also be found free in the cytoplasm. So they're either bound to the RER or they're free in the cytoplasm. So what happens on the ribosomes? What happens during translation? Well, during translation, the ribosome will move along the mRNA and it's kind of like scan the mRNA and it will translate the mRNA into amino acids. So translate the codons on the mRNA into amino acids. And that's how your polypeptide is produced. So for example, this first codon, AUG, codes for the amino acid methionine, M. So the ribosome will read that and the first amino acid will be added. Then the ribosome will move along to the next codon, AUA, and this will code for another amino acid, in this case I, and then that amino acid will be added to the chain. And then the ribosome will move on to the next codon, GUA, which codes for the amino acid V. So that amino acid will be added onto the chain, and so on. You can imagine until the entire mRNA is read and the entire chain of amino acids is produced. Alright, a further piece of information is that translation, besides the mRNA and ribosome, also involves tRNA. tRNA stands for transfer RNA. And the function of transfer RNA is to transfer amino acids to the ribosome. Right, so in order to do that function, the tRNA has a very interesting structure. It's kind of T-shaped. It has, at the bottom, an anticodon. And this anticodon is used to recognize codons in the mRNA. So imagine the mRNA has a AUG. This anticodon UAC will recognize AUG, right? So U recognizes A recognizes U, C recognizes G. So this tRNA's function is to recognize this codon, and when it sees this codon, it is to bring the correct amino acid to the ribosome. So AUG, right, codes for the amino acid M, right? That's according to our genetic code. So this tRNA's job is just to specifically recognize this codon, and it will always be attached to a M amino acid because this is its anticodon. So it needs to use this anticodon, recognize this codon, and bring the correct amino acid. Alright, so let's see how mRNA, tRNA, and ribosomes work together to make polypeptides. So in the first step, you can see that here's our ribosome, and here's the mRNA over here. So the ribosome is moving along the mRNA. Here's our first codon, AUG. Right, so the tRNA with the correct anticodon will attach to the mRNA codon. So this tRNA here with anticodon UAC has recognized this codon. So it comes to the ribosome and it attaches to the mRNA codon. And by doing so, it also brings the correct amino acid to the ribosome. In this case, M, right? M corresponds to AUG. And then the next codon is AUA. It is recognized by this tRNA with an anticodon UAU. And this tRNA with UAU anticodon will carry the correct amino acid, which in this case is this blue color one. And what happens then is a peptide bond is formed between the two amino acids to join them together. Then in the next step, the ribosome will move along the mRNA. So you can see that the ribosome has moved along the mRNA to the next codon. Right, so it's now at this next codon here. And you can see that the first tRNA is released, so it goes off, and you can actually go off to fetch another amino acid, so that when there's another codon which matches it, it will come back. Right, but the first tRNA is released, and then you can see that the next tRNA, which recognizes the next codon, will come in. So this new tRNA will fit into the ribosome. The ribosome will move along the mRNA strand, and then a new peptide bond will be formed between this previous one and the next amino acid, and so on. So you will see the ribosome move along, and then the next tRNA will come in with the next amino acid, and at the end of the day, amino acids are continually attached until the ribosome reaches what is called a stop codon. So a common stop codon sequence is UGA. UGA is an example of a stop codon. When the ribosome reaches a UGA sequence, then it knows that's the end of the polypeptide, and translation will end, and this polypeptide will be released, and it is completed. So yeah, this is your stop codon, and upon encountering it, the ribosome will then leave the mRNA, and the complete polypeptide will be produced. So in summary, genes are expressed and polypeptides are produced through a two-step process. First, transcription, where a DNA template is used to synthesize a messenger RNA. So from DNA codons, it's converted into RNA codons. And then the second step is called translation, right, where the mRNA is used to make a polypeptide. So it's translated into a sequence of amino acids. Right, so we go from RNA into a sequence of amino acids. Now you might be wondering, why is it that we have to go through this two-step process? Why can't the ribosome just go into the nucleus and read the DNA template directly and produce the polypeptide? Why do I need to make the mRNA and send it out of the nucleus? Well, one possible reason people think is that this two-step process helps to protect the master copy. So the master copy of the gene, you only have one per cell, is inside the nucleus. Right, and if anything goes wrong with that master copy, then you're done for. Right, so we need to keep it safe in the nucleus. And furthermore, right, by using transcription, you actually can make multiple mRNAs, and then those multiple mRNAs can move out of the nucleus, and they can be translated at the same time. So imagine if I only have my master copy, then it can only be one ribosome here, and then it's very slow. But with multiple mRNAs, I can actually translate multiple mRNAs at the same time and make multiple polypeptides at the same time. So it's actually more efficient in that way. Right, so we've learned about gene expression, and we want to ask ourselves the last question, which is that, are all genes expressed in a cell? Well, it turns out that actually different genes are expressed in different types of cells. So for example, in muscle cells, only those muscle-specific genes are turned on and are expressed. That's why muscle cells look like muscle cells. But let's say in your nerve cells, in your neurons, which look very, very different from muscle cells, right, different sets of genes are turned on, right, those that are specific to neurons. Those genes are turned on so that those are expressed and the cell ends up becoming a nerve cell. So it is this different, or what we call differential gene expression, that results in cells differentiating and becoming different for different functions. Okay, that's where we're going to end this. Good job for making it so far, and you can clarify your questions in class. You can also search online for videos on transcription and translation and see if that helps you.